In this fourth mini video covering the immune system, we will discuss how T cells and B cells are activated within secondary lymphoid organs, the anatomical locations in which they're activated, and we'll describe the process of clonal selection and T cell activation that leads to an immune response. So going back to our picture of our epithelial barrier that has been compromised by bacteria, I'm going to highlight again these dendritic cells. These dendritic cells have become activated by their exposure to a pathogen, and once they become activated, they migrate to lymph nodes where they can activate T cells. And so here's an activated dendritic cell that has been picking up whatever pathogen is present that caused the inflammatory response and is prepared to express those pathogen epitopes, those pathogen antigens to T cells within the lymph node. Alternatively, the pathogens themselves, if they are free-floating, or pathogen products that are secreted, can also travel from the interstitial fluid via the lymph into the draining lymph node. Now, the lymphatic system is a system for draining interstitial fluid into lymph nodes and eventually back into circulation. This is just a picture really only meant to highlight the number of lymphatic vesicles that mediate this effect. The lymphatic system drains interstitial fluid from tissues. Once it gets into the lymphatic system, we call it lymph. Lymph collects in lymph nodes along the way, and if there are foreign antigens in the lymph, those foreign antigens can activate T and B cells. And then the lymph eventually re-enters circulation via the thoracic duct into the left subclavian vein, or also via the right lymphatic duct. And so the progression goes from plasma to interstitial fluid to lymph back to plasma. This is just a table from your first aid book showing a diagram of palpable draining lymph nodes. Certainly not something that I'm holding you responsible for in this Intro to Immunology TBL, but I thought it might be interesting for you to start to look at and consider lymph nodes that might, for example, be prevalent upon physical exam. So this is a picture of the anatomy of a lymph node. A lymph node is basically a small sac of cells with a capsule. And I don't necessarily want you to know all of the terms that are highlighted in this, and so let me go through what I think is important for you to capture from this figure. Lymph arise via the afferent lymphatic and leaves via the efferent lymphatic. So this is going towards the bloodstream from afferent to efferent. Within the lymph node, you will find B cell zones. These B cell zones are located in the cortex of the lymph node. This includes primary B cell follicles that contain naive B cells, such as this B cell follicle. But also can include activated B cell zones, which are called germinal centers. And so these two gray areas would be considered germinal centers. And these germinal centers are surrounded by a mantle zone, which includes the remains of this primary B cell follicle. The T cell zone is often referred to as the paracortex, and the paracortex surrounds these B cell zones and is the site of T cell activation. It's also rich in dendritic cells. And then there are some other locations I think are important to recognize, including these medullary cords, which are rich in plasma cells. This is just some histology of the cortex of a lymph node showing a few of these structures. So for example, this would be a primary B cell follicle. These are naive B cells. Whereas here we can see fairly clearly an expanding germinal center surrounded by this mantle zone, which can include some of these naive B cells from this primary follicle. So these follicles that have germinal centers are often referred to as secondary follicles. And then these follicles are surrounded by T cells that are in the paracortex. The spleen has a very similar structure in the sense that there are B cell zones and there are T cell zones, but there are some unique features to its anatomy that you should know. First, the spleen, rather than draining tissue antigens, drains blood antigens. The bulk of the spleen is composed of red pulp, which includes red blood cells and sinusoids. We're not really going to talk about the red pulp in this session. The white pulp is what we're really interested in, and that includes the B cell zones, the T cell zones, and the marginal zone. Now, the B cell zones include primary follicles, just like the lymph node, and germinal centers of activated B cells, just like the lymph node. The T cell zone of the spleen is different. It's called the periarterial lymphoid sheath, or PALS, and it's this sheath of cells that surrounds these arterioles within the spleen. And the marginal zone is another zone kind of on the periphery of these germinal center, or these secondary follicles, that contains a lot of macrophages and unconventional B cells. And this is just a picture of looking straight on at an arteriole surrounded by this region of T cells called the periarterial lymphoid sheath. Now, how do T cells get activated within the draining lymph node or within the secondary lymphoid organ? And this is a process called lymphocyte clonal selection and expansion. Now, I mentioned previously that every T cell and every B cell each has their own unique antigen receptor, and the frequency of naive lymphocytes that might be specific for a given antigen is quite low. It's usually about 1 in a million. And so that means that the job of the adaptive immune system is to select out those lymphocytes that are specific for the antigen associated with the pathogen with which you are infected, expand out those lymphocytes, allow them to acquire effector functions, and mediate pathogen clearance. So, for example, while 1 in 10 million or 1 in a million of these lymphocytes are specific for a given antigen, if that B cell or T cell does get activated, it can undergo rapid proliferation. We call this clonal expansion. And every daughter cell from this original cell will bear the same B cell or T cell receptor as the parent cell. And so you can rapidly expand the number of T cells or B cells with the same antigen specificity. So after clonal expansion, the frequency of activated lymphocytes that might be specific for your given antigen may be as high as 1 to 50 or 1 to 20 or 1 to 10. So we're talking up to 20 cell divisions, usually within just a few days. For the activation of T cells, dendritic cells are required. Activated dendritic cells may migrate via lymph, or lymph node dendritic cells may pick up and present antigens that drain from the lymph. And these dendritic cells can interact with naive T cells, and these interactions can last for hours or even a couple of days and can induce their clonal expansion. So what happens when a T cell gets activated during this interaction with a dendritic cell? Well, this T cell needs to receive at least 2 signals. Signal 1 is the signal that's mediated by the T cell receptor. And so here we have on the top an antigen-presenting cell that expresses an MHC molecule bound to a pathogen peptide. And it is interacting with this T cell receptor. But there's some other components to this interaction that are critical. One is the presence of this co-receptor, CD4 or CD8, stabilizes this whole interaction. The last component of this interaction that I want to draw your attention to is this complex of molecules here collectively called CD3. And CD3 is a collection of molecules of subunits that congregate together, and these are the molecules that initiate the T cell activation signal into the cell. The T cell receptor actually doesn't have an intracellular domain that's capable of inducing a signaling cascade. The signaling cascade is induced by these CD3 molecules, this CD3 complex. CD3 is actually the marker for T cells. And so collectively, we call this signal 1. TCR interacts with peptide-presenting MHC, co-receptor, and with CD3 to initiate a T cell receptor activation signal. But this is not enough. You actually also have to have a second signal. And this second signal is provided by the dendritic cell. So dendritic cells that have become activated will upregulate the expression on their cell surface of a class of molecules called co-stimulatory molecules. In this case, 2 related molecules called B7-1 and B7-2. These molecules will bind to their ligand on T cells called CD28, which will then induce a survival and activation signal into this T cell. This is one way in which a T cell ensures that the response it is about to make is appropriate because this dendritic cell has been activated by a pathogen. A dendritic cell that hasn't been activated will not express these B7 molecules and therefore will not efficiently activate T cells. So co-stimulation, or signal 2, is required for productive T cell activation. Activated dendritic cells express B7, which binds to CD28 expressed by T cells. And these co-stimulatory signals enhance T cell activation and survival. So in summary, we have a naive T cell within the T cell zone or paracortex that gets activated by a dendritic cell that is presenting antigens on its MHC and also providing signal 2 co-stimulation. These T cells undergo clonal expansion and they also acquire functions, and I'm going to call these now effector T cells. These effector T cells are now prepared to migrate into tissues and mediate their effector functions. Some of these T cells, however, differentiate into helper T cells that rather than migrating out of the lymph node, migrate over to the B cell zone of the lymph node. And there they can enhance B cell activation. So in the final mini lecture, we'll discuss how B cells get activated and induce antibody responses, and we'll discuss the effector functions of those antibodies as well as the effector T cells that are produced.